Our research, creating a novel deep learning pipeline to generate and screen molecules for hormone-positive breast cancer treatment, aims to find a way to generate and screen for novel molecules that have a high potential to treat hormone-positive breast cancer through deep learning. The drug space is extremely large, and there are many molecules that have not yet been discovered that can be useful in treating breast cancer. Using neural networks makes this drug discovery process much faster and more efficient than in a laboratory setting. Our deep learning pipeline starts off with training an NLP-based variational autoencoder, similar to one engineered by Asparuh Guzik that uses a Keras seq-to-seq model and builds off of it. We trained this model with Zinc's 250,000 drug molecule dataset. We then generate molecules from this variational autoencoder by making predictions on a test molecule set. These test molecules are 68 different molecules that have been proven to bind to our desired targets. These target proteins include PI3K, mTOR, aromatase, CDK4, and CDK6. These generated molecules are then filtered to see if they pass a threshold of a quantitative estimated drug likeness score of greater than 0.6. If the molecules pass this threshold, they go to a next property prediction model, engineered by Panapatia et al. These molecules are then filtered to see if it has a predicted synthetic accessibility score of less than 3 and a predicted solubility score between 1 and 5. The solubility score is a log P value. If the molecules pass this threshold, they're then filtered and gone on to the ETOX-PRED model. This ETOX-PRED model predicts the toxicity of these molecules. And if the tox score is less than 0.6, these molecules pass that threshold and are then filtered to go on to AutoDoc-VINA. AutoDoc-VINA predicts the binding affinity between the molecule and a designated target protein by using the free energy score between the two. If the absolute value of the free energy score is greater than 6.5, then the molecule passes this threshold as well and completes our screening process, creating a final novel screened molecule. As for experimentation, we experimented on Panapatia's model with the use of an extra bidirectional LSTM layer, and we also experimented the translatability of this model by also using it to predict synthetic accessibility rather than just solubility. We can see that this model is extremely translatable because it is more accurate when predicting synthetic accessibility on an unseen holdout test molecule set. We can see that it is more accurate for synthetic accessibility for both with a bidirectional LSTM layer and without it. We could also see that the bidirectional LSTM layer makes predictions more accurate for both synthetic accessibility predictions and solubility predictions. Our second part of our experimentation is with respect to our variational autoencoder. Our VAE's decoder originally has 4 vanilla GRU layers. We decided to test the effects of replacing these 4 vanilla GRU layers with bidirectional GRU layers and bidirectional LSTM layers. Our results show that the model with the vanilla GRU layer generated 8 valid molecules from our test set, with only 2 of them having QED score of greater than 0.5 and all 8 molecules having an average QED of 0.49. The bidirectional GRU model, on the other hand, produced only 2 valid molecules from our test set, with both of them having a QED of greater than 0.5 and an average QED score of 0.78. The bidirectional LSTM model was not able to generate any valid molecules. From these results, we can say that our vanilla GRU model produces a higher quantity of molecules, while the bidirectional GRU model produces less molecules, but at a higher quality. This generation process generated 3 molecules that passed our rigorous screening process and met the threshold for all of our metrics that we explained before. The first molecule has a very high binding affinity to the PI3K P110 alpha protein, which means that it has the potential to successfully inhibit the PI3K ATK pathway that regulates division and growth of these breast cancer cells. The second and third molecules have an extremely high binding affinity to the aromatase cytochrome P450 protein. This means that these molecules have the potential to successfully inhibit the production of estrogen and progesterone, which are the hormones that are necessary to activate cell proliferation pathways of these cancerous cells. Our experimentation allowed us to create a drug generation pipeline that allows us to find molecules that have the potential to treat breast cancer. This pipeline is extremely translatable and can be used not only for breast cancer, but for other diseases as well. This architecture also has a lot of room for improvement, as others can also add to it and also use it in various other drug design contexts. Thank you.